Good morning children. I am Sarita Isis. Now I am going to teach you transforming principle or Griffith's effect. Let's see. Frederick Griffith conducted an experiment on Streptococcus pneumoniae. So Streptococcus pneumoniae is a kind of bacterium that causes pneumonia. And he was conducted an experiment on this bacterium, Streptococcus pneumoniae, and he observed two strains of this bacteria. The strains are S strain and R strain. These are the two strains of this bacteria. The S strains are smooth strain. They are smooth and that is virulent. Virulent means harmful, that cause disease. Virulent. And it causes pneumonia disease and it forms colonies, smooth colonies in a culture media. Smooth colonies are formed. Smooth colonies. Okay, so the S strains are smooth, they are virulent and they cause pneumonia disease and they form smooth colonies in a culture media. But the R strains are rough. They are rough in nature, they are avirulent. Avirulent means it's not harmful. Avirulent and do not cause disease, pneumonia, and that form rough colonies in the culture media. Rough colonies. Okay, I'll show you the strains. These are the two strains of Streptococcus pneumoniae. The smooth strains, and one more peculiarity is there. In the smooth strain, there is a capsule covering made of polysaccharide. That is, a polysaccharide cork or capsule covering is there. But in case of a rough strain, there is no capsule cork or capsule covering. That we can write over here. So, capsule covering present. But here, in rough strain, no capsule covering or no coating. Okay, so these are the two strains, and we can see the experiment conducted by Frederick Griffith on these two strains of Streptococcus pneumoniae. Okay, next we can see the S strain. He injected the live S strain. Live S strain is injected into a mice. And he observed that the mice died due to pneumonia. Because the S strains are virulent, that cause pneumonia disease to mice and that cause death of the mice. And in the second step, he injected S strain, that also live S strain into mice, injected into mice. And the mice live. Okay. And third one, the third step, this is the first, second. In the third step, the heat killed S strain. Heat killed S strain. What is the meaning of heat killed S strain? That is, the S strain bacteria can become avirulent due to heating. So when it heats, it becomes avirulent. So there is no positive organism or no virulent bacteria when it heats. So the heat killed S strain, when it injected into mice, there is no pneumonia. So the mice can live. Okay. And in the fourth step, he mixed the heat-killed S strain with R strain and then injected into the mice. So most probably when the R strain is injected, there is no symptom of pneumonia because it is avirulent. And like that, heat-killed strain also, S strain. Here, S strain. Heat-killed S strain also avirulent. So there is no chance of pneumonia in this case. But you observe that the mice die due to unexpected symptom of pneumonia. Actually, what was happening there? Here, from this dead mice, he recovered live S strain or S cells. Okay, actually here the S strains are heat killed and there is no live S strain plus R strain. There is an unexpected symptom of pneumonia that caused the death of the mice. From the dead mice, Frederick Griffith recovered live S strains. So actually what is happening here? He concluded that the heat-killed S strain caused the transformation of R strain into S strain. Once more, the heat-killed S strain caused the transformation of R strain, or we can say the transformation of a virulent strain to virulent strain, S strain. Okay, so this phenomenon or this process of transfer of one character or a trait from one bacterium to another directly is called transforming principle. Okay, so this is transforming principle or Griffith effect. Griffith's effect. Actually, what is Griffith's effect? The process of transfer of characters or traits from one bacterium to another bacterium directly. That is called the Griffith's effect or transforming principle. And the transformation of this R strain into S strain is due to the presence of or due to a genetic material. But Frederick Griffith fails to explain the biochemical nature of this genetic material. He fails to explain which is the actual genetic material that caused the transformation of R strain into S strain. So next we can see the biochemical nature of transforming principle. Now we can see the biochemical characterization of transforming principle. As I told you, Griffith fails to explain the biochemical nature of genetic material. And the scientists such as Oswald Avery, Colin MacLeod and Maclean Carty, they repeated the Griffith's experiment to find out the biochemical nature of transforming principle. And they purified the biochemicals such as proteins, RNA, DNA, etc. Carbohydrates also from the S strain, heat-killed S strain. So, from the heat-killed S strain, they purified biochemicals such as protein, DNA and RNA to find out which is the genetic material that caused the transformation of R strain into S strain. So we can see three biochemicals are purified from this heat-killed strain. Now we can see what will happen. The first protein which is taken from or which is purified from the heat-killed S strain is mixed with the R cell. So there is no transformation of R cell into S cell. is the observation. And second, the RNA from the heat-killed S strain is mixed with the R cell. There also no transformation. Okay. And finally, they have taken the DNA. The DNA already purified from the heat-killed S strain is mixed with R cell. So there is a transformation of R cell into S cell. And they are using a DNA digestive enzyme known as DNase. They add DNase in this along with the DNA. So what will happen? R cell in R strain. So the DNase can digest DNA. Actually, DNase are enzymes which can hydrolyze or which can digest DNA. It is a DNA digestive enzyme, that is DNase. When the DNase digests DNA, there is no chance of transformation. Okay, so here no transformation. That means the transformation is inhibited by the DNase. Okay, likewise, they added protease. Protease is an enzyme that can digest protein. Protease plus DNA plus R cell. Transformation is there. Transformation. Why? Because protease digests protein. So the protein do not affect the transformation. If DNA is there, it can transform R cell into S cell. So transformation is there when they were added protease. Protease is an enzyme that can digest protein. Another, they were added RNase plus DNA plus R cell. There also transformation occurs. That means the RNase can digest RNA. So if there is no RNA, in the presence of DNA, the R cells can transform into S cells. Here, from this experiment, they were concluded that the genetic material is DNA. So DNA causes transformation of R strain into S strain, and because of that, the mice died. So this is the biochemical characteristics of transforming principle or Griffith's effect. Hope you understood everything about the transformation principle and the biochemical nature of the genetic material. That's all for today. Thank you.